Thank you so much for the invitation to come chat as part of the Environmental Studies Lunch and Learn seminar series. I'm a big fan of the program. I teach courses in sort of the food systems track in environmental studies. And I often don't get to talk about what we do in our lab here at Tufts. I usually go out and tell other people, but I rarely get to tell the Tufts community. So I'm very excited to be here today to tell you about what we do in the lab. And so the way I'm going to start this talk is by first explaining to you what microbiomes are. And these are very important microbial communities, collections of bacteria, yeasts, filamentous fungi, viruses that live everywhere. So this room has a microbiome. Some of the food that you're eating has a microbiome. You all have a microbiome living inside of you right now that will digest that food later on today and into tomorrow. And what we've been learning through all the exciting science of microbiology over the last couple of decades is that these are ubiquitous systems and they're essential. So microbes are everywhere and they do a lot for life on our planet. Everything from training your immune system to growing all the food that we eat. Everything that you eat every day has been touched by microbes in some way, either through the benefits that microbes give us or through some of the things they do that are kind of annoying as well. And I teach a course here called Microbiology of Food, a non-majors biology course that you can take if you'd like to every other fall usually. And what we do is we tend to think about the roles that microbes play from production of raw food materials, like growing corn, for example, or even growing a microbe, mushrooms, through the processing of food materials, everything from distributing food and keeping food clean and maybe even adding preservatives to maybe more traditional things like this kimchi party that's happening right here, processing kimchi, all the way to your gut. So we really think about the roles that microbes play from farm production, processing of food, and ultimately to the food that ends up in your stomach. And I think this is an important context to set before I start talking about what we do in my lab today, because my lab's research really falls here in the middle. We think not so much about the production side of things, and we do collaborate with folks to think about what happens when you eat food and it eventually gets to your gut. But what we're mostly going to be talking about today is this part of the food system, processing food in the middle. And mostly we're going to be talking about fermentation. Okay, so a little bit about what microbiomes are. They're everywhere and they're super important for everyday life. And it may seem like maybe we've got everything figured out about microbiomes, but actually there's a lot we don't know. And I'm going to tell you about some of the really big questions that we're still trying to understand about these interesting microbial communities that are everywhere. And one of the big questions that a lot of microbiologists are asking right now is simply, what are the parts needed to build a microbial community? You can kind of think about it like if you're trying to build a car on an assembly line, there are particular parts in a car that need to go into a particular place at a particular time to get a fully functioning car. And what we're trying to do in the field of microbiology right now is ask the question, what are the microbial species, the parts, that need to live in a microbiome, whether it's inside of your gut, whether it's inside of a cornfield, or whether it's inside of any environment where microbes live? What are the parts needed to get a healthy, functioning microbial community to do what you want it to do? How do they fit together? What are the design principles or the assembly manual, if you will, for these microbial communities? And we actually don't have really good answers to that broad question right now. Another really important challenge in microbiology is almost all of microbiology, almost everything we know about microbes has come from studying just a few microbes living all by themselves in a petri dish in the lab. And so as I've been saying, microbes live out in the messy, complicated communities in nature, but almost everything we know about microbiology comes from this environment, a petri dish, very simplified environment with usually just E. coli or maybe one or two other microbes living there. And as you could imagine, and if you take an ecology class, you know this very well, that when a species is living by itself, versus when it's living with other species, different members of the same type of microbial community, or even very, very different organisms, things change, right? There are species interactions that can dramatically change the biology of microbes. And so one thing my lab is trying to do is move from this monoculture thinking in microbiology to thinking about the more diverse communities that exist in actual natural systems. Another big question that we've been trying to answer in the field is what generates microbiome diversity. And I really love this figure from an early human microbiome study that was done by Julie Segre's lab at the NIH. And what they did is they went out to a bunch of healthy individuals and they sampled body sites. They took swabs and they sampled body sites everywhere from up inside of people's nose all the way down to between their toes. And that's what we're looking at here in these pie graphs. Don't worry about the complicated microbial names, just look at the different colors. And the different colors represent different types of microbes in those habitats. And hopefully what you can see is that up in your nose versus on the palm of your hand versus between your toes, you have very different microbes living in those habitats. So we can answer these questions. We can make these beautiful microbial landscape portraits, if you will, quite easily. But then answering why do we see variation in the diversity of microbes across the human body or other habitats? We don't know. We actually don't have great answers to it. In the human microbiome, one reason we don't have great answers is it's just hard to do experiments with microbes on humans. Imagine trying to do a study where you manipulate the microbes between your toes, right? Lots of challenges there of getting that experiment to work properly. And then a final big question, a big challenge in the field of microbiology and microbiomes is how do we take all these answers that we're seeking, all this knowledge that we're trying to gain, and apply it to benefit humankind? And these types of applications are happening out in agriculture. There are many companies right now that are essentially trying to make probiotics for corn, for soy, for wheat, and other big crops that we grow to maybe grow them more sustainably through the power of microbes. And then another very big area that we'll briefly touch on later is the gut microbiome. And this is, again, this massive microbial organ that you carry around with you that does so much for your body every day. And there are many companies right now trying to work on how can we engineer and manipulate that? How do we figure out what are those parts in your gut microbiome? How do they fit together? And how can we change that when things don't go right? For example, when you have a digestive disease. Okay, so that's sort of the place that my lab and our interests and my teaching also fall within the broad scheme of biology and science. And you may be sitting in there saying to yourself, okay, Professor Wolfe, those seem like pretty simple questions, right? Why haven't you all figured that out by now? This seems pretty straightforward. And there's actually been quite a few challenges in microbiome science that have prevented us from getting very solid answers to some of these questions. One is simply that microbes are generally hard to culture. So the top left, I'm showing you some petri dishes from my lab. We're looking at various molds and bacteria growing there. We try really hard in microbiology to grow all the microbes we can, but most of them are hard to tease into a petri dish and grow really easily. And we have a saying in the field, if you can't grow it, you can't know it. And so it's really hard to understand the biology of something that's hard to grow in a petri dish. So that's one big challenge. Another big challenge is the challenge of complexity. So most microbiomes have hundreds or thousands of species living in them. So just like a puzzle that has 10,000 pieces is much harder than a puzzle that only has 10 pieces, it's really hard to take apart all the parts in these complex systems, understand how they fit together, and then put it back together again. And so the complexity, simply the sheer species diversity in most microbiomes has been a big challenge for pushing through and answering some of these questions. Another fascinating sort of philosophy of science or sociology of science kind of issue is that much of the field of microbiome science initially had research silos. So people that studied fungi were in departments of mycology. People that studied bacteria were in bacteriology departments. People that studied the gut microbiome would be at a medical school, and rarely would all those people talk to each other. But microbiome science requires all those people to talk to each other because the challenges that we're facing are interdisciplinary challenges. And so what's been happening is those research silos have slowly been dissolving and going away, and we're all starting to work together in an interdisciplinary space. The last thing I'll point out is that it's generally hard to get to a lot of microbiomes, not all of them. And the ones we'll talk about today are super easy. You just have to open up a jar of kimchi and boom, they're there. But if you study the gut microbiome or even the soil microbiome, getting to where the microbes live can be quite challenging. So accessibility has also been an issue. Okay, so what is our solution in our lab? What do? We essentially take a model systems approach. So just like many scientists study the mouse as a way to understand human biology, what we've been doing in my lab is studying simple systems, like mice are simplified versions of humans, or zebrafish is a simplified version of an animal as well, or Arabidopsis is a simplified version of plants. We take these model systems and we pull them apart and put them back together again. And hopefully we can learn generalizable principles we can take out to more complicated systems. And in our case, our model systems happen to be delicious, which is also very convenient. So we are using things like kimchi, sauerkraut, kombucha, sourdough, and cheese as model systems for microbiome science to really be able to answer some of these big, challenging questions about what are the parts and how do they fit together, but in a system that is pretty simple. So generally speaking, when you make kimchi or you make cheese or you make tea, and we'll talk about this later on, there's only a handful of species, 2 to maybe 5 or 10 species of microbes, as opposed to hundreds or thousands. And so it just makes it much simpler to be able to take that system apart. The other thing about the fermented foods that I'll talk about today is they're everywhere. People are making these all over the planet. So like little microbial islands that we can go out and sample, and people are doing them in replicates all over the world. Another thing that goes back to 2 slides ago is that almost all the microbes that live in fermented foods, we can grow in the lab. We can get them to grow in a petri dish quite happily. So that solves the if you can't grow it, you can't know it problem. And then the last thing I'll point out is that what I love about working with fermented foods is that they are inherently interdisciplinary. It involves chefs, it involves food producers, it involves microbiologists, it involves farmers. It brings all those people together through the lens of microbes to understand how do we build microbial communities. And so I think it's really the perfect solution to this grand problem that we have in microbiome science. So what I'm going to talk about today is what are we doing with these delicious fermented foods and what are we learning about them? And what I'm going to tell you about are 3 ways that humans shape the diversity of fermented food microbiomes, sort of 3 broad things. And again, if you've taken an ecology class, some of these things will be quite familiar to you. The first thing we're going to think about is what are the parts that are available? What are the microbes that can actually get to your fermented food of interest, whether it's kimchi or cheese or whatever happens to be? And that is a process that we call in ecology dispersal, right? So we can easily see birds flying over and getting to a pond, or we can even see seeds of acorns from an oak tree moving from one place to another. It's really hard to see microbes dispersing, right? When was the last time you saw like E. coli walking by and going somewhere else? You rarely see that. But what we're trying to do with fermented foods is understand the microbial species that are out there, the potential species, and then the species that actually get to the habitat of interest, that land in the place of interest. So that's the first thing we're going to think about today, dispersal. The second thing that humans do to shape the diversity of fermented food microbiomes is they play around with, once they have their parts, how they fit together. And this is what we're going to call interactions today. So just like squirrels interact with an oak tree by moving acorns around, planting them, and you get future oak trees, microbes interact with each other in various ways. So microbes can help each other, they can support the growth of neighboring species, or they can inhibit each other. They can actually compete and duke it out with each other on wheels of cheese and elsewhere. And what we can do in the microbiome space is use those interactions to dial up or dial down particular species of interest in a system. And the last thing that humans can do, and we've actually been doing this unintentionally for many, many years, is we can work with the process of evolution to change microbiomes over time. And this is something that my lab is actively working on. And what do I mean by that? Well, imagine if we take this microbiome that we have right here with these 3 individual species, and we happen to put it out into 3 different environments. Maybe one of you, when you go home for fall break, you take it to California, another person takes it to Minnesota, and somebody else takes it to France. Let's just say that's where you happen to be heading. In those different environments, those microbes will experience different things, different temperatures, they'll experience different maybe salt conditions, maybe you'll have different amounts of light coming in. And so the environments will be different in those places, and that could lead to the process of natural selection, causing evolution to unfold in your fermented food. And so this is something that we're really interested in part because humans, by chance, have been playing with the evolution of microbes over many years to domesticate them to grow in fermented foods. So from the yeast that we use to make bread to fungi that are used to make miso, humans have been evolving various species of microbes in these fermented foods. And I'll talk a little bit about that today as well. Okay, so how are we exploring these two? What are the systems I'll talk about today? One is, I'll talk about sourdough. How many of you have a sourdough starter by any chance? You know, it's hard to maintain one in college, but we have some sourdough lovers here. That's great. So what a sourdough starter is, that I'll talk about in a bit, is simply wet dough that you grow bacteria and yeast in to make more interesting bread than Wonder Bread, right? Our typical white bread that we often eat is just a single yeast, but sourdough is a diverse community of microbes that are used to make bread. And the story I'll tell you about there is that the story of San Francisco sourdough, how it's somehow special and unique in the grand world of bread, it's not really that special from a microbial perspective, and we'll talk about why. And the second thing I'll do is, once we have a bread course, if you will, we'll put some cheese on it and talk a little bit about how microbes living in cheeses are fighting with each other and also helping each other as well. Okay, so let's go to the sourdough. Obviously, during the pandemic, sourdough had a really big moment as we were all locked away, running out of yeast. Everyone started making sourdough at home, at least it seemed like that. What I love is how different parts of the media explored sourdough interests. Some people, you know, like CNN, New York Times, or even the CBC thought it was lovely, whereas Fox News was talking about it being a wasteful and difficult process. Everyone has their own opinion about everything from, you know, the world to sourdough. But it was definitely something that a lot of people were doing at home. And so what were they doing? What were they actually doing? How are humans working with those microbes? Well, what a sourdough starter is you take flour, you get it wet, and you let microbes grow. It's as simple as that. And the goal is to get a specific type of microbial community to form. And ideally, it would involve having yeasts, lactic acid bacteria, and acetic acid bacteria. And you may not be familiar with these names of these microbes, but you've already interacted with them in your life. Yeast makes bread. Lactic acid bacteria, anyone that ate yogurt today, you had a big old spoonful of billions of lactic acid bacterial cells. Those are the tangy things that live inside of yogurt. And anyone know what acetic acid bacteria make that you've probably interacted with on a regular basis? Any guesses? What do you put on French fries sometimes to make them taste really interesting besides ketchup? Vinegar. Acetic acid is vinegar. And so acetic acid bacteria make vinegar and a bunch of other metabolites that we use as humans. And so by this community of microbes living together in wet dough, when you go to leaven the bread, when the bread starts to rise and make all those bubbles and eventually gets baked, the microbes are living in the wet dough, breaking down the dough and adding flavor. So again, if you made Wonder Bread right next to sourdough bread, the sourdough bread is so much more complex, has a more interesting crust, has a really depth of flavor profile. It's just so much better. And that's because of this community of microbes. The sour in sourdough comes from the lactic acid bacteria and acetic acid bacteria making acid. Acid is sour, and so the sour tang of sourdough comes from those microbes. So when we started this project, it was actually before the pandemic made sourdough popular. What was really interesting to us is even though people have been making sourdough in the US for many decades, it's been very popular here for a long period of time, almost all the research about what microbes were living in sourdough came from Europe and just came from a couple of bakeries, a few studies here and there. So all of the sourdough starters that people were buying from King Arthur or were sharing with each other, we had no idea what yeasts or bacteria were living in those. Were they the same as what was in Europe? Were they different? What were the communities like? So that was one really big thing that we were curious about. And so we teamed up with an amazing network of folks at the University of Colorado Boulder, as well as folks at NC State, to do a large-scale survey of the biodiversity of sourdough starters. So we asked people all over the world, send us your sourdough. Put your sourdough starter in a bag, put it in an envelope, send it to Tufts, and we're gonna sequence the microbiome to understand what microbes were there. And when we started this, we thought we maybe would get like 50 or 100 sourdough starters. It turns out we got 550 of them, which became a bit more than we expected. The mail person kept bringing up the mail and being like, what are you guys doing? Because they had no idea what all these stinky packages that were arriving at our lab were all about. And you can see this is an amazing undergrad Stravia that was working in the lab with a subsample of all the envelopes of sourdough that got shipped to the lab. You'll notice that most of the samples came from North America, and that is because most of the shipping was easiest from North America. It was just logistically easiest to do that. But we got some samples from Australia, we got some samples from parts of Europe as well. So we really were able to do a deep dive of what's living in the sourdough of North America. Okay, so how do we sequence a microbiome? And this, what I'm going to show you right here, would be true for anything, everything from your gut microbiome to an agricultural microbiome. In this case, I'm illustrating it from cheese, which we'll talk about later, but this would apply to any microbial system. What we do first is we get the genetic material, the DNA, out of the microbes. We have kits that can extract that DNA out. And the next thing we do is we purify that DNA in different ways. And if anyone's interested in this technology, we can talk about it later. There's different fingerprinting genes that we can sequence. And there's these tools called shotgun metagenomics where you can sequence all the DNA. Once you have your DNA and you've purified it in some way, the third step is you have to sequence it. You put it into a machine, a DNA sequencer. These are the same sequencers that were developed for the Human Genome Project back when that was happening. And then the third to fourth step, this is the biggest jump here from 3 to 4. This is a challenge. And if you're in computer science or have friends in computer science, tell them to go get a job in this in the future, because there's a lot of bottlenecks here. You have all this DNA, but then you have to figure out what is it, right? What are the species of microbes that DNA came from? And so what you have to do is take those sequences and match them to databases. Try to say, how does the DNA that I pulled out of my fermented food match existing DNA sequences in databases? And that's a big, grand challenge there, how to match those two things together. And at the end of the day, what you do is you get a profile back that hopefully accurately represents the original community of microbes that was there. So that's overall how we do that. And I can answer any questions you have about that technology later on. And then this is what you get, a very busy, very colorful graph of the microbiome of a particular sample. And again, this is for our sourdough project. And what I'm going to do is walk you through how to understand one of these microbiome profile graphs. So on the top, in these colors up here, the top row, that's all data for bacteria. So those are those acetic acid bacteria, the vinegar bacteria, and then they're also data for those yogurt bacteria, the lactic acid bacteria. On the bottom, we have data for yeasts. So the yeasts, which are the main gas producers, the things that make the bubbles and give structure to sourdough, we've got the yeasts on the bottom. And every column here, every really thin column that you're seeing in this graph, that's an individual sourdough that we sampled. Remember, there were over 500 of them. So there's over 500 lines here. And what I want you to do, don't worry again too much about the scientific names that we have here. Just know that different colors, as they're getting bigger and smaller, mean that different sourdoughs have unique species living in them. And so if you're just kind of looking at this broadly, are all sourdoughs the same? No, right? You see a huge amount of variation. So for example, over here in this part of the data, we have this kind of beigey color, and we have this like, I don't know, I call it cat puke brown, but I don't know what color that is to you. It's like a greenish mustardy color. And that's a community that we kept seeing over and over again. So those are many sourdoughs that had a bacterium called Lactobacillus sanfranciscensis, named for San Francisco, or was originally identified. And they have the yeast Saccharomyces cerevisiae. Anyone know where you might find Saccharomyces cerevisiae? Ever hear of that yeast before? Sourdough, but also where else? Yeah. Sorry, what was that? In brewer's yeast, yeah. So in beer, excellent. Beer, wine, and other fermented beverages, you often use Saccharomyces cerevisiae. We also see it in sourdough as well. So that's one community type, but you'll notice there's many different types of communities. So we have this red and blue pattern over and over again with Saccharomyces cerevisiae. That's a different set of bacteria. We have even more diverse sourdoughs over here. So this is where I love this part of the data set. There's all this variation, right? Every sourdough looks different in that part of the data. Okay, so this is step one, going out and just really sequencing a lot of samples and saying what microbes are out there, what's out there. The next step is the why, right? Why do we see all this variation? Why do we see some that only have 2 species? And why do we see some that have a ton of diversity in them? And that's what the lab's been doing ever since we did this huge sequencing effort. And so what we did is we come back to our sort of model of the 3 different things that happen that humans interact with to shape the diversity of these microbial communities. And again, it's dispersal, microbes getting there, it's interactions, microbes fighting with each other or helping each other, or it's them evolving over time. And one of the first things we thought about is dispersal of sourdough microbes. And the reason we thought about that is this idea, again, the story of San Francisco sourdough, that when you buy sourdough in San Francisco, there's something special about it. It tastes different than sourdough elsewhere. It has a different texture, it has a different appearance. And people over and over again suggested that there are unique microbes in the Bay Area that make San Francisco sourdough special. And you could imagine why that might be. And this is, again, a basic ecological idea. And this is that in San Francisco, maybe there are unique microbial communities that don't leave San Francisco. They don't disperse from San Francisco. Maybe the climate there, the fog rolling in from the ocean, the very mild temperatures selects for particular microbes that can't live here in New England, where it's really cold and we have wild fluctuations in temperature. And this is an idea in ecology called dispersal limitation, that you get unique communities of plants, animals, or microbes simply because they just can't get everywhere, right? They land in a particular place where it's ideally suited for them and they don't move all over the place. And so one of the things we wanted to test with this huge data set is this idea of dispersal limitation. Is there sort of a pattern across our data set to prove that San Francisco sourdough is somehow special? And we had to make a graph to prove that, to test that idea. And I just want to show you what it would look like in the ideal world. And this is getting into a little bit more advanced ecology, but it's pretty straightforward here. So if we expected there to be dispersal limitation, that there were unique microbes everywhere you go across the US, we could make a plot of the geographic distance between every sourdough that we sampled, right? We could just measure how far apart each sourdough sample was. And the dissimilarity of the community of microbes. So what I mean by that is that if the community of microbes was exactly the same between two sourdoughs, they had the exact same species, the dissimilarity would be very low, right? They'd be identical, so it'd be very low on the y-axis. But the more different the communities get, the higher up on the y-axis you go, and you get this nice plot where the further apart you were, the more different the communities were. Does that kind of generally make sense? It's something that's a very common thing that people look for in ecology. So let me show you what we found. A hot mess. We found a lot of dots all over the place with very little signal of this positive relationship between the geographic distance between samples and how similar those communities were. And we kind of expected there to be a lot of noise, but we still expected that maybe, again, if there was dispersal limitation, we'd see some kind of pattern. But then after thinking about it more and more, we were like, wait a second, maybe this makes sense. And the reason it makes sense is that people are moving sourdough microbial communities all over the US all the time, right? When I would move from one part of the country to another, I could take my sourdough with me. Or people can ship sourdough. You can go on Etsy and buy a sourdough starter right now from Texas or from Hawaii or from Alaska. And so humans, again, this is how humans are shaping these microbiomes. We're moving them around all the time. So there really is no strong geographic signal simply because humans are swapping these microbiomes all over the place. And so there isn't anything unique, microbially speaking, about San Francisco sourdough. There could be other things people are doing in San Francisco to make the bread special, but there's nothing about the microbes that makes them special. Okay, so that's one thing that we thought about. The other thing that we wanted to sort of understand, again, if we go back to our big pattern of diversity, we can't explain it with dispersal. Like, we don't see that all of these microbial communities over here came from California, for example. So what could be an explanation for all this diversity? Why do we see the communities that we have here? So the next thing we thought about are the interactions. How do the species fit together? How do the microbes essentially talk and play with each other in these delicious sourdough communities? And a very pioneering student in the lab, a PhD student, Liz Landis, what she did is she took all of the main microbes that you find in sourdough, all the main yeasts and all the main bacteria, and she grew them together in pairwise combinations. She said, if I put them together and give them a chance to sort of figure each other out, what happens over time? Do some things win? Do some things lose? Does everyone live together in happy coexistence? And this is in the lab. So this is outside of the more complicated sourdough world. This is where everyone kind of has an equal chance when we mix them together. And she developed this really elegant system for studying these in the lab. And what she found was, I think, something super fascinating. So what I'm showing you on the right here, all these different colors are, again, the same dominant sourdough microbes I showed you earlier. We've got both the yeast and the bacteria. And what I'm showing you is the relative amount of each of the interacting partners after they were transferred together for many generations. So over 6 different transfers that Liz did. And you'll notice, like, for example, here in this row, where we're mixing the yeast Wickerhammermyces anomalous with a bunch of other yeast, you'll notice that the Wickerhammermyces, which is this dark brown color, almost always wins. It really just outcompetes all the other yeasts. But we often have peace. We often have coexistence in these systems. And I think that's actually very fascinating when we have that. So for example, this red and blue we have here, the Lactobacillus plantarum and Lactobacillus brevis, they're really happily playing with each other. And there's many other examples of coexistence of these different yeast and bacteria with each other. And when we start looking at what we saw in the lab, and then back out to the real world, this data set, we start to see a lot of interesting repetitions of the coexistence. So for example, one of the most dominant things that we see co-occurring is the red and the blue. Uh-oh, hello, come back, there we go. Oh, giving it away. This red color and the blue color represents these 2 bacteria. We kept seeing those over and over again in real world sourdoughs. And those are the 2 that most commonly got together in the lab and happily coexisted. So it seems like humans are doing something to encourage the peaceful coexistence of these microbes in sourdough. And again, could explain some of the variation we have in this data set. I will tell you, we looked at everything from the presence of a cat in the house to the type of flour that was used by these people. We had a huge metadata matrix, and it didn't explain very much at all. There's just a lot of variation in what people are doing, and I think there's a lot of variation in what the microbes are doing to each other in these systems. And so we still have some more work to go to really explain this diversity. Another question you could ask, that's really cool, Professor Wolfe, interesting basic ecology, but I really care about the taste of sourdough, right? I care about how sourdough tastes. Can you tell me something about that? So we can step back here and say, okay, we've got lots of diversity, lots of variation in the communities. Does that matter for how the sourdough is going to taste? So we did that. We worked with some cool collaborators in chemistry, and Liz also did some really interesting experiments where she set up these test tubes in the lab, and she scientifically measured how quickly the dough would rise. You can see this is a time lapse over about 24 hours. And different sourdough starters that are in each of these tubes had a different rate of how quickly they would make those important bubbles that create the great structure in sourdough bread. So that's one thing we did. And then with our chemistry collaborators, we measure what are called volatile organic compounds. Those are the aromas, the smells of sourdough that make the bread really delicious. And we can measure those because the microbes are making them. We can trap them into various pieces of equipment and measure what those particular properties are. And again, another really busy microbiome graph is about to appear in theory. There we go. And so this is one of the main things we found about how the variation in the diversity of the microbes correlates with the function, the things that they're making. So what I'm showing you here is, it's called a heat map in microbiome science. The blue color means there's not a lot of something. And when you start to get like yellow and red and orange colors, there's a lot of something. So that's the first thing to know. The other thing to know is this is a list. So each row here is a different volatile compound, different chemicals that we were able to measure through this chemistry collaboration. And one thing that we noticed when we looked at all the columns being a subset of all the sourdoughs that we were looking at. So these are individual unique sourdoughs that we studied in the second part of the study. We started to notice that there was a big division between these sourdoughs, so all of these columns, and these sourdoughs over here. There's a very distinct difference in the volatiles that they make. So for example, a lot of the butanol compounds and some of the alcohol compounds, these are things that are sort of fruity aromas or sweet aromas. There was a lot more in all of these sourdoughs compared to these. And the biggest difference between those starters was the presence of those acetic acid bacteria, those vinegar-making bacteria, really explain this big division between the broad patterns and the functions of the sourdough. Even the dough rise rate was explained by whether those acetic acid bacteria were there. And what's super cool in the world, at least of sourdough science, is that in most studies of sourdough starter microbes, people have ignored those acetic acid bacteria. They're hard to grow. They're hard to, unless you're using the DNA sequencing we did, you don't really detect them. And so our study showed that these vinegar-making bacteria are really abundant in sourdough and might help explain why your sourdough has the properties that it does. Questions about where they come from, where are those vinegar bacteria coming from, and how do you get more of them? Those we don't have answers to yet, but it sort of does help explain some of the functional properties. So from our sourdough story, what we learned so far is that San Francisco does not have anything special about it, microbially speaking. That the interactions, the way that microbes interact with each other might help explain some of the variation. And these vinegar bacteria are really widespread and may play important roles in the properties of your sourdough bread. And what I want to do in the remaining few minutes I have is just give a shout out to some of the cheese work we're doing. This is something we've been doing for a much longer period of time, and many of the current projects in the lab are focused on. And when I talk about cheese, I'm not talking about your classic sort of plastic wrapped cheddar. That's a very wonderful and important cheese. But what we're talking about here is artisan cheese is often what it's called. And they're also known as surfaced ripened cheeses. So think of camembert, right? With that strange fuzzy white rind. It grows microbes on the surface, which then ripens the cheese. The delicious rot and the title of the talk is because the microbes are slowly decomposing the cheese, giving it that sort of stinky funk that you get in a lot of these types of products. And there's many different types, everything from camembert to things like cloth-bound cheddars that we call a natural rind cheese. And these are just some of the microbes that we see in the cheeses. They're really beautiful. This is growing them on a plate in the lab. And you can see right away, without knowing much about microbes, the diversity of forms and shapes that they have there. And if you look at this even closer, so the rind, that layer on the top is what we're interested in. It's a great gig because we sample the rind and we eat the paste. We don't really care about the paste, so we get to eat it in the lab, which is really lovely. And it's this community, just like in plant or animal ecology, where we have a succession. We get a mixed group of microbes that grow over time, living on that surface of the cheese. And this is a process that's happening all over the world. Just like people making sourdough, there are small artisan cheese operations all over the planet, everywhere from Argentina to China to here in the United States, making these products. Everything from getting the milk to shaping it into particular shapes and eventually aging it. That's where the rind will eventually develop. These are some of the places that we've worked at in the past. Our best collaborators, people that we work with a lot, are up in Greensboro, Vermont, up in the Northeast Kingdom, almost into Canada there. This is Jasper Hill Farm, really a pioneering artisan cheesemaker in the United States. And they've been a great partner in exploring the microbial ecology of their cheeses. And that's very high tech. We've been in a very low tech situation in a very small town in Eastern France. This is the mayor and the local cheesemaker. He has multiple jobs. And he makes this funky looking cheese that was delicious. It looks like it's gone bad, but this is exactly what he wanted, this very fuzzy looking cheese. And I'll point out the thing in the top of his cave, those are not cobwebs, that's a fungus growing, these strands growing down from his cave, which is under his barn. So this is what, again, cheese development would look like if you were walking into one of these particular environments. So just like the sourdough study, we went out and sequenced the microbiome of these cheeses. We wanted to know what's living in these products, in part because at the time, most of the work had been done in Europe because these are relatively new products in the United States. And we made yet another graph, and we used the exact same approach. So I'm just going to show you this again. We went from DNA all the way to one of these microbiome profiles. And we got, again, another nice microbiome profile. So hopefully you can use what we learned about how to interpret the sourdough one and look at this one. And again, the answer is there's lots of variation, right? There's many different types of rind microbial communities that can develop on these products. And some of it is being driven by the humans. Again, how humans shape the microbiomes is by what they do. So for example, if you're a bloomy rind cheese lover, the camembert types of cheeses or brie and even the goat cheeses from the Loire Valley, they have a very distinct microbiome type. And this is because the cheesemakers are adding particular cultures and doing particular things to the cheese. And that we find in that part of the data set over there. But there's still a lot we don't know about what's in these particular products. We still don't know what they're doing with each other, how they're helping or hurting each other. And so that's, again, the microbial war and peace that we've been doing in the lab. And one thing that we do in the lab is we dissect the microbiome and put it back together again. And this is an example of how we do that in what we call in vitro cheese. These tiny little 96-well plates are like this big. And each of those little dots there is a unique cheese rind that we're trying to mix together to understand how these parts fit together. And there's a very cool example in this one that you can visually see. What we've done here is we have different rows of fungi. So the rows going across are different fungi, and we've mixed them in pairwise combinations with different bacteria. And one thing you'll notice is right here, see how this is much lighter compared to every other circle? And that's because this bacterium, Arthropacter, is inhibiting the growth of that fungus. So we can actually see an example of war where the bacterium is actually fighting for metals, really scarce metals, in this case, magnesium. And so we do this over and over and again, and we can learn about the ways that microbes interact in this system. And I don't have time to get into it because I wanted to mostly focus on the sourdough, but these are just a couple of the ways that we're learning about how microbes fit together in the system to make a bigger microbiome. Everything from microbes producing antibiotics in cheese. So when you eat a piece of cheese, there might be a little bit of a future antibiotic there that we have yet to discover, to microbes living on each other in a way that almost looked like a superhighway, where the fungi would grow out, make hyphae, and the bacteria would hop on like an HOV lane and cruise around on the surface of cheese. To very recent research in the lab where you smell cheese, those aromas that we can detect are microbial signals, ways that they can actually talk and interact with each other. This is, again, just an overview of some of the work we're doing in our lab. If you're interested in this, if this is at all fascinating to you, I'm happy to chat more about what we do, and you can come to our lab and see some of this in person as well. Okay, so just a quick recap. We're using fermented foods as model systems to go from the microbes to the food and understand how these different parts fit together. And it's really mostly basic science. Most of what we're doing is just trying to understand how do microbes work. But along the way, if you can figure out the design manual for a microbiome, you've got some useful knowledge, right? You can do helpful things. And so one of the parts of my job that I love the most is when we get a Franken cheese sent to the lab. So this is cheese gone bad. Something's gone wrong here. It could be dispersal. It could be interactions. It could be something about evolution. And we put our detective hats on. We go in and try to figure out what's going on. And a really cool project that was started by an undergrad was trying to understand, why did this cheese turn purple, which is not a color that a cheese should be. In this case, it was a bacterium that got into the system. So dispersal was playing an important role. And it was making a precursor to indigo, the stuff that we use to dye blue jeans, which turns out to be purple. So the cheese turned purple because of this indigo compound. And we have this happening all the time in the lab. And it's a really cool way to interface between Tufts and food producers throughout New England and the rest of the world as well. The other thing that I absolutely love about fermented foods, and this is a bit of an advertisement, is that we can use them in the classroom. It's a cool way to get students to learn about microbes through a highly relevant system that you have in your everyday life. So everything from Bio 106 and 107, which is our majors microbiology lab and lecture, where we adopt fermented foods and we use them throughout the lab to learn principles of microbiology, to when I teach Bio 108, which is sort of every other fall, and that's the microbiology of food course. And we also do outreach beyond Tufts. So we love to go out and tell the world about the wonders of microbes. And this is something that we heard about earlier. We used to have a farmer's market booth where we go out and have microbial cultures and cheese and give away knowledge. We don't have food samples, but we give away the knowledge about microbiomes as well. And as you heard earlier, we also do a lot of outreach sort of online or in the media. We do have a free website, microbialfoods.org, where you can download like salami microbiology posters or other random stuff about microbes and fermented foods. And you can also check out our work in National Geographic. And as we heard earlier too, we're also in this really interesting show called Cooked on Netflix. And with that, I will thank the many people, the many undergrads that actually, and grad students and postdocs that help this work come to life, as well as funding from mostly the National Science Foundation, but also the USDA as well for supporting this work. And if we have time, you can ask some questions about microbes. Thank you very much.
See a lot of collaborations here between the Cheese Club and the Wolfe Lab.
Yeah.
Maybe questions. I can get us started. I was trying to wrap my head around all these wonderful interactions, and my mind kept going back to flavor. And I was wondering, in your opinion, what is more important, or to what extent is more important, the substrate or the players on the substrate?
It's an interesting question. So both in our sourdough study and in our cheese study, one of the things we looked at is what was the substrate? So what kind of flour? Was it all-purpose flour? Was it wheat flour? Was it rye flour? And in the cheese study, in that big sort of rainbow of data, we also asked about, is it goat milk versus sheep milk versus cow's milk? And it turned out that in terms of the microbial perspective, in terms of them assembling into a community, it didn't really matter. So you could find identical microbiomes on sheep's milk versus cow's milk versus goat's milk. For flavor, it completely matters what the substrate is. So you can imagine when you eat a goat's milk cheese, there's that classic goatiness, right? That flavor that you get that some people don't like, but is also great for some other people. Versus a cow's milk cheese won't have that flavor potential to be broken down by the microbes and released. So I think that the substrate matters a ton. Once you get within a substrate, you can start to play with the microbial community, and that's where you can get even more variation. So in all of these fermented foods, there's all these different layers of what is the raw input in terms of the food, what is the microbial community, and then also the levers that people use to turn up and down those different things. Salt, how much you add, when you add it, the temperature of fermentation. But yeah, it's a great question. I think that the substrate is hugely important for driving flavor at the end of the day. Yeah.
Thank you. And
yeah,
while I get there, I'll ask you a follow-up.
Sure.
You just started mentioning something about the different levers that people use. Are there other mechanisms that people can use to change the communities and in turn change the flavors? Have you encountered this in your interactions with farmers or food producers?
Yeah, so I think dispersal is one of the most important things. So what you allow to get into the system can potentially completely change the flavor profile. So we saw that purple cheese at the end, which is a bad example of dispersal. Essentially a weed got in and started to make the purple color. But in the production of sourdough starters, one thing that people can do is you can often add fruit, or you can sort of mix in external things that aren't necessarily part of the flour. And the idea there is the fruit has yeast on it. It has microbes on it that could eventually intermingle with the microbes you already have and make a more complex community. And there's an undergrad in the lab asking questions about that right now. If you start mixing together different microbiomes, how do they eventually form into a community, and what's their function as well? Yeah.
Hi, so it seems like in both of these cases, you're showing that there's like lots of different bacteria and yeast growing on these things. We don't really know a lot about them. So I guess my question is like, what makes us confident that all of this is like okay to eat? Like, why does it make us sick?
That is
and like, when is cheese gone bad? I guess.
Yeah. So I will say that in both our sourdough study and our cheese study, we didn't detect any pathogens, any known, like listeria, for example, which is a really important pathogen in food systems. So we don't see a lot of widespread contamination. There are sometimes outbreaks, just like in romaine lettuce, which is always seeming to have a bad day. But cheese sometimes has a bad day too, and you do sometimes accidentally get listeria in some of these products. It can happen, but it's not very often. But I would agree that I talk to a lot of cheesemakers and they say we're essentially blindfolded. We're sort of making solid milk after you go from liquid to solid. You let microbes grow, and generally it works. Generally it's safe. And I will point out that for cheese production, they do a lot of safety testing to look for pathogens. They may not know the full microbial profile in every batch of cheese of the good microbes, but they can definitely say for sure that they don't have listeria or they don't have Staph aureus or any of the bad things. So that's just part of like modern food safety testing. What we're trying to do right now is develop through a USDA grant, develop rapid diagnostic tools. So essentially like a COVID test where you can quickly tell if you have COVID or not. It'd be awesome for these cheesemakers to dip a stick into their cheese and know exactly what microbes they have and be able to track that over time and correlate it with some of the sensory properties that they detect. But that's like a 10 years down the road kind of thing. Yeah, it's a good question. Thank you.
Hi, so in some other classes, genetic diversity has been a hot topic of, I guess, worry of genetic diversity declining rapidly, especially I like read up on how it's been declining in human populations over, the centuries. And I was curious on your take on, I guess, looking at the gut microbiome diversity and how that is being affected by this loss of diversity.
Yeah, it's a really important question of, as we take antibiotics and a lot of the other modern things that we do, because of the wonderful advances of medicine, how is that affecting the microbial diversity we have in our guts? And then what is the functional impact of that? And so one way I can kind of answer that is through a collaborative study we did where we tracked participants that ate fermented foods and looked at whether if you eat fermented food diet, how does that change the microbial community you have inside of your gut? And fermented foods have been around for hundreds of years in many cultures. And the idea is that they're both nutritionally good, but also maybe deliver a microbial benefit. And what we found is it had a huge impact on the gut microbiome. There's a dramatic shift in the composition of microbes in your gut in terms of diversity. What we're still trying to figure out, and groups at Stanford and groups in the University of Wisconsin and other places are trying to figure out, is what does that mean from a health perspective? And so one of the questions is, if you have a fermented food diet that you're eating and you're getting some of those microbes, does it change your immune system or does it help you digest food better? And there's some signals that it can reduce inflammation. You can actually have, if you have a high inflammation gut microbiome, which can cause like leaky gut problems or sometimes various digestive issues, some fermented foods can actually suppress that inflammation and help with those issues. But it's really early on in those kinds of studies. In general, though, I would say that as in many communities, having more diversity is generally thought of as a good thing because you have sort of a backup plan. You have a extra species that can have extra functions that could buffer from various types of perturbations. But there's still a lot we're trying to figure out in that space. It's a great question. And as a follow-up, I'll say that all the identities of the microbes that I gave today really at the species level, but there's a whole bunch of levels below that at the strain and like higher, like more resolved genetic diversity that I didn't talk about that we're starting to measure as well. Yeah.
Sort of building off of that, can certain fermented foods like cheese make your gut health worse? I know a lot of people say they have adverse reactions to cheese, and we get a lot of requests about lactose, but I don't know if that's related.
Well, the nice thing about many of the cheeses that we study is most of the lactose has been eaten up by the bacteria. So if you are very lactose sensitive, some of the long-aged cheeses are actually generally fine. But you're right that some people can be sensitive to some of these cheeses for a variety of reasons. Some people have sensitivities to fungi. Like if you're in a very moldy environment, or even if you eat mushrooms, some people have allergic reactions. So if you eat a wheel of camembert where there's lots of fungus, that could be a problem for you. The other thing that we're beginning to understand in the US in our food supply in general is this thing called biogenic amines. These are these compounds that microbes make as they're decomposing food that some people can essentially have anaphylactic shock. You can have it's almost like you're getting a cold all of a sudden and you start sneezing and then you start to swell up and it can be really problematic. And in some of these foods, because of the way that certain microbes decompose the cheese substrate, you can have a high amount of those. And it's just not on the radar of the FDA or other people right now. But in Europe, they're starting to really regulate those kinds of things. But yeah, it's a great question. There can be negative health effects of some of these foods. And you have to think about the costs and benefits, right? If you're getting a probiotic bump, but maybe there's you sneeze a little bit after you eat cheese, like what's the cost there? And how does it all wash out? Yeah.
Thank you.
Yeah. Yeah.
It's 1 o'clock, but thank you so much, Ben.
Of course. Thank you all.